My name is Joseph Kim. I'm the president and CEO of Inovio. It's my pleasure to introduce Dr. David Weiner, um, considered to be the father of DNA vaccines, one of the key pioneer in the field of DNA medicine. David, it's great to be with you here.
Joseph, it's wonderful to be here with you.
So I have some questions about DNA vaccines and DNA medicine. So how did this start?
Well, that's a really interesting question. When we started this several decades ago, really there were two technologies for making vaccines: live approaches or dead approaches, or non-live. And live approaches really were our favorites because by growing inside our bodies, the immune system learns to fight off the infection, and also because the virus actually replicates and produces proteins inside our bodies, we can not only make antibodies to the soluble proteins that are produced, but also our killer T cell compartment, the Navy SEALs of the immune system, are engendered, which can then expand and contribute to viral clearance and better protection and control. And so that was the hallmark of live infection. However, live infection can take very long to make and be safe and has lots of other developmental issues. Non-lives really have a whole safety advantage because they're non-live, but they lack many of the immune functions of the live approach. And so we were thinking if we could deliver a non-live, non-replicating, completely controlled piece of DNA and code the instructions, the cells of the body would make the proteins. They would, again, stimulate an immune response antibodies by being shed from the cells. And because it's also made within the cells, it would have the hallmark of induction of killer T cells. And because they're non-live and can't spread, they would actually have the safety, therefore, of the non-live. And so you'd wind up with the best of all worlds. And also, DNA is really simple to manufacture.
So what do you think is the most unique character of DNA vaccines?
Well, that's another really important focus that we have been on and along this journey collaborating with Inovio. The sequences themselves are not derived from any organism. They are designed by computer, and there are algorithms that Inovio has now developed that enhance this design and make it very rapid and very reproducible. It's also the vectors themselves are reduced to practice. The manufacturing system is very consistent, and the delivery has been simplified, and the formulations have been improved. And so this is a local delivery for a local expression only in the skin or in the local arm, no production other parts of the body, that can produce proteins that can be seen by the immune system and stimulate a strong immune response as if they're an infection, and also generate very consistent levels of CTLs in this very reproducible, consistent delivery platform.
So David, you and I have been working on DNA vaccines for over 25 years. So a lot of people ask me, what makes mRNA different from DNA and vice versa? And how would you compare and contrast two different technologies?
Well, mRNA and DNA are both genetic material, but they have very different purposes. DNA is kind of the library of information. It's very stable. It can be extracted from extinct animal systems from thousands and thousands of years ago. And that is just a credit to how stable it is. And one piece of DNA can make many, many messenger RNAs when it gets into a cell.
So I believe the main difference between DNA and mRNA vaccines in people are DNA's ability to generate, as you mentioned, CD8 positive killer T cell responses, and mRNAs have not. Can you explain why that might be so and what people have been seeing in the clinic?
Well, right now we have very little DNA, information about RNA in the clinic. There's only been a few studies reported, and of course, we're learning more and more. But what has been reported is they can, with multiple injections, drive T cell responses, but it seems like they're biased towards CD4. The DNA seems, which we have a lot more data for, a lot more published data. There's, especially in the clinic, is driving both antibodies and CTLs.
So, what concerns you most if you look at the DNA vaccines, um, and also looking similarly at mRNA vaccines? Well, from a scientist and from a vaccine developer's perspective, what would be your greatest concern, and what could be the greatest positive from those two vaccines?
So I think we're in a unique time that nucleic acid approaches such as DNA and RNA are being considered and moved to the forefront, and we're on display through the CEPI funding for COVID. They were the first two vaccines that were moved forward in the United States, for example, through CEPI funding, and they were both part of the first three groups that were funded by CEPI. So that's quite an interesting thing, and that speaks to the advantages in concept these have for moving into the clinic rapidly, delivering something people believe in a simple, rapid fashion, and also possibly providing benefit. DNA, we have had several indications in the clinic of the first appearances of efficacy. The treatment of HPV-induced cervical neoplasia, the double-blind placebo-controlled trial that was published in Lancet a few years ago showed efficacy. Early effects in GBM have now been reported out of Harvard that this was having some effect, although we haven't hit a true efficacy endpoint yet. And immune responses in infectious diseases that for emerging infectious diseases that look very, very similar to what we might need and might be associated with protection, but we haven't. We have more work to do. RNA is still earlier. We've seen some reports. We've seen a flu report from Moderna, and particularly, and then we've seen some other public reports, but those papers are not out yet. There have been some reports from other groups as well. However, so we have a lot more that we would like to see there, and it would be great to have more demonstrations. So I guess what I would say is on the RNA side, it would be really important to see more demonstrations of something that we can read and get our handle on and understand. On the DNA side, since it's further along, I think we are very confident about the safety and the reproducibility of that, and that there hasn't been significant adverse events. But now expanding those and moving over the goal line is really the most important thing in the DNA space.
So, speaking of nucleic acid vaccines, there are other vaccine platforms like the viral vectors and protein and adjuvant vaccines. Can you give a little bit of pluses and minuses of your thoughts on those platforms as well?
Well, viral vector vaccines are our historic favorites. They're the kind that we made measles, mumps, rubella that most of us have taken around the planet. The newer ones are really recombinant viral vectors we're mostly hearing about, and those are really, for the most part, unlicensed technologies as well. But most of those, there have been a lot of development, and there have been slow, and that is because the live vaccines have advantages in safety of these recombinant vectors over the historical vectors, and that they can't just replicate on their own for the most part, the ones most people are developing now. But their boosting and serology issues have to be really delicately worked on and tiptoed around. So they have advantages in that they can be made in large amounts, but their ability to be boosted and re-administered and things like that require a lot more work. The non-lives, we've really seen the heyday of recombinant protein, I would say, plus some of the newer adjuvants. And these are very exciting. And those tend to give antibodies and really very little or very, very poor CD8 T cells, and actually weaker CD4 in most cases. And so they've been particularly advanced in certain infections where it's a single type of antibody response that's most associated with protection. However, there's a lot of safety work and all that is built around them as they're developed because of the combination of the two platforms, the adjuvant that stimulates immune response and the building of the protein and having it stay in its right shape if that's important. And so there's work in those, but those are very exciting and important approaches. And of course, it's sort of a hybrid of those that's conjugate vaccines, which is the fusing of sugar molecules to proteins, which is also very important.
So, David, back to what we are talking about in this webinar, from DNA medicines, from COVID-19 to GBM. What really gets you excited about all of the potential that Inovio and the collaborators are going to demonstrate with respect to the DNA medicines in the clinical setting, both for the vaccines for COVID-19 as well as an immunotherapy for GBM?
So I can say that it's a very exciting time for the DNA field. We've spent, as you know, we've traveled this road together. It's been a very focused and accomplished, but step-by-step journey to build the platform and get it to the point where we start to see the amount of consistency we see now in the clinic, and the number of people vaccinated goes up to the thousands without significant adverse events, and the reproducible induction of antibodies and T cells in multiple trials, as well as the impacts now in cancer, the first impacts we're seeing, likely mostly due to the cellular immune response that is being generated, the T cells that are CD8 T cells that are being generated, that not only are induced in the periphery, but go find the tumor, turn a cold tumor hot, so-called, when it's filled with CD8 T cells, and then can eliminate or control the tumor. I mean, these are very dramatic things that are now being reported in different papers that have been through peer review. And so, and you had very prestigious groups like David Reeder's at Harvard report on the GBM data, which is a very difficult tumor that requires generating T cells that can cross the blood-brain barrier and still have effector function, still kill. That's a very unusual thing. And so I think we're in sort of a new phase of DNA, a phase of expanding what we've already accomplished. We now see we can get responses very consistently. We kind of have established a very broad number of different diverse types of people who have received these vaccines with both disease conditions such as very serious like cancer, as well as healthy individuals, so that we now really understand a lot more about what our platform can do and how consistent it can be, and seeing responses across them. And so I think we're kind of in a new age. We're in an age where DNA can start inducing and living up to its potential, that potential we've been thinking about for a few decades now, and really cross that barrier and provide us a really new and exciting tool for both vaccine immune responses and in the immune therapy arenas.
Yeah, absolutely, David. So lastly, what do you think is the future for DNA vaccines?
So that is one of, that's a very important question. What is the future? So DNA is an enabling technology for delivery in vivo and local production of biologics that are customized by each person that receives them. And they're a chemical that we're full of, that we're used to getting exposed to, that is very natural, and really is the instructions for making something. And so some of the other exciting things besides this are, of course, the induction of biomolecules as reported with the DMAbs and the new cancer targeting self-assembling bispecific targeting reagents that the patient would make themselves, which would really open up to many more patient populations access to these type of drugs. And then self-assembling molecules that do many other things one could use, as well as drug therapies themselves. So I think the vaccines and the immune therapies are an enormous thing, but they really are the scratching the surface of where we can go. And I'm going to be just so excited to see where this field goes and where we can go over the next 15 to 20 years. It should be a very, very interesting and exciting ride.
Yeah, David, I agree with you. I think we can change the face of medicine using this platform and really maximizing all of the gene products that we can deliver using this platform, from vaccines to immunotherapies to mAbs to BiTEs and bispecifics, therapeutic proteins, and I believe there's an amazing future lying ahead for this technology and this platform. So I'd like to thank you for your time and really being the true pioneer in this field, sticking with this from the beginning and really starting to see the fruits of your labor and our collective labor going forward. So thank you very much, David.
Well, thank you, Joseph. It's been an honor to be part of this group.
Great, thanks.